What are the two types of variation?
You have continuous and discontinuous down.
We have two types, continuous and discontinuous. Right. Now, discontinuous, they're like discrete variations. So with this one, it's like either you have a certain trait or you don't. For example, blood group. Tongue roll. So either you roll your tongue or you can't. You have a specific blood group. With the continuous variation now, you can get a range out of it. So the continuous variation is the one that has a range. So like for height, foot size, right? Anything that can give you a range, which means that, go ahead.
Fingerprint, would that be continuous too?
No, tell me why it wouldn't be continuous. Fingerprint would be, would more than one person have the same fingerprint?
No, so that
No.
is continuous.
Right.
But it can be in the same category. Like
All right, could you plot a histogram for fingerprint?
no.
All right. So if you can plot a histogram or a bar graph, you would put that under continuous variation. And right, so if you're doing a tally, well, I shouldn't say it like that. In terms of, so like with the histogram, it would be for continuous variation. Discontinuous, it would have to be a bar graph. So if you can plot a histogram with the trait, it's continuous variation. If you can plot a histogram, anybody remember how histogram work?
Yeah, you have to have different intervals.
They have intervals, right? And how the bar graph work?
It does have like, it does have different categories, and if you're in the
Right, exactly. Just like what we said for discontinuous variation, right? You have a particular category and you're either in it or you're not in it. So quick example, ability to tongue roll. All right, so number of persons. All right. This could be the ability to roll the tongue. All right, so let me just put
pie chart.
Pie chart, sure.
Yeah.
No, I just want to show that with the discontinuous, you're placing them in distinct categories. So what any form of chart that shows distinct category, you can use it. Clear? I don't know. I would say this is thumb rule and no thumb rule, or however. So with discontinuous variation, they are placed in distinct categories. With continuous variation now, so like you would say in terms of feet size, right? So let's say 4 to 6, and you would have that. And then like from 6 to 8, then from 8 to 10, something like that. So with continuous variation, you will have a range. Do you understand? I'm not sure.
Yes, sir.
All right. Now, we need to look at what leads to variation. So if you understand, do you understand the word variation in terms of biology? What does the word variation mean?
All right, the difference in the trait of an organism.
All right, within a species, right? So
Yeah.
there will be differences between individuals in a species. It can be due to what? Two main things or two main factors that cause variation.
Meiosis.
No.
Well,
Well,
no, wait.
Yeah. All right, genetics and the environment. All right, so kind of genetic changes that leads to variation. That is the most important one, or the biggest factor. The change, once you have changes in the genome, then you will have variation occurring. All right. Now, if we look at our, all right, let me just clear this. Looking at what causes variation. Let's put that on the board. So it's environment and it's genetics. When it comes to genetics, it is mutation. We are changing up structures within the DNA. All right. So what causes variation? Environmental factors. For example, what? All right. Let's say I have an identical plant, right? And you put two plants. Identical. Okay, just a second. If you have two identical plants, right? And you place one under ideal conditions. And the other one under harsh conditions. So remember, we start out with two identical plants. What we are doing here is looking at how environmental factors can lead to variation. So if you have two identical plants starting out with, and you put them under different conditions, will they look the same? So like, will they have the same, will they go to the same height, same width in terms of their leaves? What will happen? Will they look the same? That's the question.
No.
So
So right, there might be what? Change, variation in height.
Sir, what about color?
Right, so variations may occur in height, color of leaf, the size of the leaf as well, right? size of leaf. Let's say it's a fruit tree, right? The size of the fruit can also be affected, right? Size of leaf, size of fruit.
Sir, and one can bear fruit and maybe one don't bear none.
Probably that too, right? So the question now, the height, the change in the height and the color and the size, right? It would probably be due to the amount of nutrients, water, and so forth. Question, would this impact the gene in the plants? Did some change in the genome occur? Or it's more physiological?
Sir, if it's the identical plants, I don't think that's a good change, only like the physical features, right?
Right. So the physical features are changed. Good. So then, if the plants reproduce, right, would that affect the future, the offspring, in terms of the genes the offspring is getting? Is the offspring getting the same gene or different gene?
They're the same gene because it wasn't altered. It's just
Wasn't
like
altered.
right.
Right, that's the point. The gene wasn't altered, all right? It's just physiological changes, so it never grew. If it had gotten the nutrients, it would have grown properly, but the genome was not altered, all right? So what is this suggesting? If the variation is in like height, size of leaf, color, and fruit, right? Could we put that under continuous or discontinuous variation? These features,
It can go under continuous.
it would go under continuous. So when the environmental factors causes variation, it is not influencing the genome. So the type of variation it leads to would be continuous. All right. So environmental factors, it's not affecting the genome. So it would not lead to any discrete phenotype being formed. What if now we change the genome? So mutation, what is a mutation?
Will affect the makeup of
The
the
gene.
gene.
Right, once you interfere with it, you cut it, you add it, flip it, do something to it. Once you change it, it mutates, all right? The mutation, it can be a single base, and when you interfere with it, it can have serious implications. All right. So it's not like with the environment where it don't have enough water, so it grows short. It can be like sickle cell. One, you interfere with a base, and the shape of the red blood cell, it change. So with mutations, that one is more serious, all right? And it can lead, and that one, it leads to discontinuous variation because you're actually now interfering with the gene. Right, let me just clear this. So we can have mutations. We can have mutations in a gene, which is the main mutation there, point mutation. Anybody notice a point mutation? A point mutation, the change so it's just a single change in a nucleotide. And an example under this, you have sickle cell anemia. It's just one change, and it basically messes up everything. One simple change. All right. So point mutation, a change in a single nucleotide. Now, the chromosome can also have mutation. Anybody know of them? The type of chromosomal mutation, insertion, deletion, remember that?
Oh, yeah.
Translocation,
Translocation, right?
location, inversion.
Substitution.
Give me a second. So we can have, repeat them for me.
Insertion,
Translocation,
substitution,
duplication,
Sir, isn't insertion
Point.
gene mutation? Isn't it
On the
inversion?
point.
I'm not hearing you.
Yeah, hear me now? Not sure.
Yeah.
Repeat about the insertion if it's a point mutation.
Yeah, I'm asking if insertion wouldn't be gene mutation and inversion would it be the chromosome? Because inversion are when you like switch the position, but insertion are when you put in a whole new one.
Mm.
If you understand what I say.
Yeah. I'm going to do an example of each of them quickly. All right, the names kind of, for some of them, the names will actually give you a little hint. So you can add, you can delete. So if it's delete or add in, you have what we call frame shift. You use the term frame shift before? No? All right, so if you're adding, all right, let us say let me not say ABC. Let's use actual. So AGG CU, right? A. Let us say this is the normal genotype, right? Or the normal sequence of nucleotides in the gene. If it says insertion, what do you think we should do? For insertion. The name explains itself. Insertion. What are we going to insert?
Nucleotide base.
Just right. So this is your normal. The abnormal one would arise from a mutation. So if it's insertion, just put something else in there. So let's say AGG. Between the G and the C now, we just add a new letter. And then you continue with CUA. All right? So you're adding a nucleotide for the insertion. What would be deletion?
yeah, delete one.
Exactly. So this one that we just did was insertion. All you're doing is just put something. So for the normal one, the abnormal one, it has something new. Question. All right. This is the normal sequence, right? AGG. If we're making an amino acid, how many bases do we read each time for the amino acid? If you're making a protein, all right. So AGG, that would be one amino, right? So AGG, that would make one amino acid from transcription, translation stuff. The next amino acid would be CUA, right? No. What just happened here? Yeah, you will have your AGG, right? But will you still make the amino acid that CUA codes for?
No.
Right. Since we're talking about amino acid, let me just use a base that is actually in. So AGG, you make amino acid. Will you make the next amino acid CUA codes for? Yes or no?
No.
No, right, because it shift out of frame now. ACU. So looking on, let us say AGG, a glycine, and CUA, a lysine. In this DNA, you're not making lysine. You're going to make a different amino acid. So now you have a different amino acid, which means that whatever is being produced is going to have a different function, which is the case in the sickle cell anemia, because they switch out something, a different protein, a different amino acid is going to be made. All right, so look, just by inserting a different base, so once you change one of them, you're making a different amino acid here, which will cause a problem. Because remember, proteins, the sequence of amino acids in a polypeptide chain, and then it fall upon all that. If you don't get the right amino acid sequence, the protein is going to change, right? So just by adding one base or removing it, the amino acid that is made is going to be different, all right? So let's do deletion. Let's random example, CGAA, CC, all right? A. All we need to, so this is the normal one. All we need to do is delete something now. Normal, abnormal. So let's go with CGAA. Let me replace. No, I need to delete something. All right, so look at this. So you have GC, CGA, ACC, A. With this one, CGA, ACA. What was deleted?
One of the C.
One of the C. All right. So in these, the name is helping you to know what type of mutation is occurring. With the inversion, as the name suggests, probably not so well. Give me just a second. Let me just. All right, insertion. Version. Go over this, please. Which part? The deletion? Talk quickly. All right, so for deletion, you will have your normal gene, right? You delete something. In this case, I deleted one of the C's. That will cause the abnormal gene. All right? So that is all we're doing. Deleting something from the gene. So this is the normal one with the two C's. If you have a gene with one C instead of the CCA, you just have CA. So something is missing. That is with deletion. Right? With insertion, you add something that was not there before. With inversion, this is on the chromosome now. So let's say I have 2 chromosomes. So there's a portion of a chromosome, and next portion is here. Let's say I have A, G, or AC, A. Right, ACG.
It's
All
the
you're,
inversion, right?
right, all you're going to do now is to flip it. So ACG, it becomes G up here, C and A, right? So it's just the opposite. So just flip it 180. So ACG, GCA. Give me 1 second.
Sir, we never to get the insertion one.
All right, read it again. What are you saying now, Shaniko?
I didn't to get the insertion one.
Insertion?
Yeah, the insertion.
It just adds something to the
In
normal
the
sequence.
abnormal or the normal?
No, so you have the normal one, right?
Yeah.
For it to become abnormal, you need to do something to the normal one. So if you notice I add an A in red here, so it's no longer the normal one. That's the mutation. But
Okay.
once you do something to the DNA, it's a mutation. All right, so the normal one, AGGCUA. In this one, it having an extra A. That's a mutation. All right. In this one, I remove the C. It delete. That's a mutation. It's not the same as it was before. Down here now on the chromosome of this sequence here, ACG, but on this one, it flipped, GCA. So it's inversion. So on this one, ACG over here, CGA. Clear?
Yes, sir.
All right. For duplication, and then we do translocation.
Sir, so the one in red is for inversion?
These two are inversion. It's showing
Yeah.
inversion. Yeah.
All right.
Oh, yeah. Let me leave. Inversion. All right. So for duplication, let us say I have AGGU. Let me just color. We're just focusing on the letters. Right.
Sir, you can maybe bring it up further because I can't really see at the bottom.
Oh, all right. All right, so if it says duplicate, what does duplicate mean?
Make adjustments.
Like, yeah, make a copy, like,
Right.
Make
So
a
you're
copy. Yeah.
right. And you're making an exact, right? And so ACCG, right? So this is the normal one. All right. Abnormal. All right, so we have ACCG. If we duplicate it, you're going to have A, C, C, G again. All right, so that's all. You have 2 copies of it. All right, so that's duplication. Anybody have an idea what translocation mean?
Sir, like one of the letters them that is supposed to be somewhere fixed, it's gonna be like somewhere else.
Right. So we move it and attach it somewhere else. All right, just so this we did was duplication. So with duplication, you're just duplicating a specific portion. Insertion, you're flipping it. Deletion, remove something. Insertion, you add something. Translocation, we're going to remove it and put it somewhere else. All right, so let's see if we can do that here now. All right, so we're doing translocation. All right, so ACC. I'm going to cut out ACC, all right? So this is normal. Hold on. All right, CCG. So CCG, ACC. So I move the ACC from up here and put it down here. The translocation, you're taking a portion of the gene from one end of the chromosome and putting it somewhere else on the chromosome. All right, so it's just movement. Take it from here and put it at the next position. We understand this?
Yes, sir.
All right. So this was translocation. All right.
Sir, them switch positions, but we're not really seeing the bottom part.
Oh, sorry about that. I forgot. Yeah. Did you see the normal one in green, the ACC? Oh, can I see this one here? Are you seeing it?
Yes, sir.
All right. So, right. So the ACC in the abnormal one, just put it in front of this or behind it. Doesn't matter. Just move it from here and put it somewhere else.
Okay.
Yeah. So translocation, it just means take it from where it is and put it somewhere else. Do you know how Down syndrome come about?
Or extra something.
Hmm?
I like when I'm about extra something.
Yeah, extra chromosome,
Chromosome. Yeah.
right? All right. We are going to that part now. Sir, we need to reach. You're Gucci, you know. All right. No, we can't do that. All right. All right. So to explain, I want to explain two types, the Down syndrome. To explain Down syndrome, you have a term, it's called non-disjunction. And if you're familiar with this term,
No, sir.
all right, so it will occur in either, it can occur in mitosis and meiosis, all right? So what it will lead to is the offspring having less or more chromosome. So you know that some can have an extra chromosome. All right, so let us show you. Click. Some people have an extra X or one, like some female, if you have 2X, they have 1X. Or some persons have 2X and a Y. So in some cells, all right, in your gametes, right? How much the gamete, is it haploid or diploid? I shouldn't say it like that. After
haploid.
meiosis, yeah, at the end of meiosis, it is haploid, right?
Haploid, yeah.
Right. So after meiosis 2, you will have 2 haploid cells, right? So let us say this one is from a female now. We're looking at, so after meiosis 2, to form the gamete, to form the zygote, we would have fertilization, right? So let us say this is a haploid cell from the female, haploid cell from the male. And the two of them fuse, you would get the diploid offspring, right? So let us check how we come about now, where some cells have 3 and some will have 1. So we're doing meiosis, right? So you know that in meiosis, in the first division of meiosis, do we have haploid cells or diploid cells? After the first division,
Yeah, that.
right? So meiosis, first the division. So remember it has 2. How many division does mitosis have?
1.
One.
Right?
Oh, one. Mitosis.
Mitosis have 2. In the first division of mitosis, do we have chromatids in these cells or chromosomes? As in, do we have homologous chromosomes or a single strand of chromosome after the first division of mitosis?
Homologous.
All right, should I have, is it this?
No.
Or is it
So
this?
there's a
If
mitosis and meiosis.
Meiosis after the first division. I'm skipping out metaphase, anaphase, telophase, but straight to cytokinesis where it split up. They start
Yeah.
to, yeah. So I'm saying at the end of the first division, should the 2 cells look like this? That is what I'm asking.
Yeah.
Yes. Because what would have happened between this diagram and this diagram? What would have happened? Which stages are missing?
Metaphase, anaphase.
All right,
Telophase.
just a second. So this is prophase, right? Right, so I was asking what happened between this stage and this stage. Just as synopsis. The chromosomes will
bit-
align here.
Align at the
Equator, right?
equator.
And then what happens after that?
Then
Them I go start get
Them
pulled
a-
apart.
What pulls them apart?
The microtubule.
The microtubule.
Tubule.
Spindle fiber.
Right, that's
Spindle
what I
fi-
said.
Sorry. Then what is the wait, alright, never mind.
Yeah, ask what is the spindle fiber made of?
No, another one there.
All right. Right. So between prophase
Sir, what's the difference between microtubule and
And spindle fiber. Come and think them are the same.
the camera on here, Shanika.
And it,
No, all right. Microtubules are like the apparatus then that make it. You have your spindle fiber, right? But what is it actually made of? That's the difference, as in what make it up. But a revision, so we can't go too much into that. Right, what I was expecting you to tell me is quickly that they line up at the equator, the spindle fibers attach at the centromere and pull the whole homologous chromosomes apart. Right, but if you don't remember, let us just do it quickly. So, after prophase, what should happen in this cell? What should it look like?
the
It
third
is metaphase.
pull them apart. It will look like a two different cell line.
it's a guy in a middle to metaphase.
Oh, I
Right.
think that's up on a face, sorry.
No, yeah, metaphase. So the chromosomes would be in the middle of the cell, right? Does the spindles attach it?
No.
No.
Sure? No
Yeah.
attach?
Yeah, them for dip on them.
Right,
Yeah, at equator.
right.
Oh,
So by the time we reach anaphase, what happens now?
They're
Them
being
I get
pulled
pulled
to
to
the
the
end of
opposite
the
poles.
yeah.
Them.
Right. And it's a kind of bend, right? So,
Yeah, like that. But you would have to have the two of them.
right, I'm gonna try, yeah. I'm gonna do this.
They
Yeah,
just
it's a
try to
bend.
X them.
Yeah. I'm gonna try to, yeah, it bend, all right, that, yeah. All right, yeah, see that. Yeah, something like that. All right, I have the spindle fibers attached. No, the thing is, all right, so just like how this is happening, right? And then we know that these cells are going to do what? They will eventually split in two, right?
Yeah.
Yes, sir.
And then one is up here and one is in here. Non-disjunction means that this did not happen. They did not, for some reason, it not separate. All right. So this would be the normal process, all right, of meiosis 1. So this is meiosis 1. This stage is anaphase. Now, let us look at the non-disjunction. The non-disjunction is that these two chromosomes, they did not separate. What will happen? One of the cell will be empty and one of them will have what? Two chromosomes
them.
inside, right? All right. So this is where the problem will start, because when this cell undergo meiosis 2, what will happen?
No,
I don't
no,
want
no.
it to-
No, no,
All
take
right.
place.
Right. So if the cell divide, will anything be inside of them?
No, sir.
So,
No. Let me see if I can explain everything on this board. All right. So let's say this is from a male, right? So let us focus on this. What I'm explaining here is non-disjunction, where the chromosomes, they did not separate. The diagrams in red is what should happen. This one in blue is non-disjunction. They are in the same cell. They did not separate, all right? So if they did not separate, one of them will be empty. Good? Now, let us take female haploid cell. Let me use a different color. Just follow me here on this. Right? So the female, select it's an egg. So should two things be in there or one? If it's an egg,
One.
one, because it haploid, right?
Yeah.
So right, so this combine with this empty one now, because we're the fertilization. You will get what? One chromosome instead of two, right? Pay attention. As
Yes,
in
sir.
you understand?
Wait. If this one, start from right this one.
You have to follow, right? This right here, the cell, the chromosomes did not separate into the two cells. One of two of them, and the next one of none. So this one without any. If it fuse with an egg cell, remember it don't have any, you know. So if it fuse with it, this person now, the offspring here, is going to be, will it have extra chromosome or fewer chromosome?
Fewer.
Fewer. An internal syndrome, an X chromosome is missing. So we are showing you how a cell can happen to have a chromosome missing. All right, so non-disjunction, it can make a cell have more chromosome or less chromosome. So you see for these now that have none in it, so understand this one where we get the one. You see the other cells here, they should have three. The end result is that one of them will have three and the next one will have one. Try slowly. All right, let me see if we can do that again. All right, so before I move on, everybody understand this, right? We get the one and the zero.
Yeah.
Yes,
All
sir.
right.
Yes, sir.
Right, so we're gonna look now how we get three in one cell. All right, so let us focus on this one now, which means I'm going to erase this part. All right. So when this cell divide now under the second meiosis, how much chromosome will be in each of these cells?
Oh.
Two, all right. Remember, after meiosis two, how many chromosomes should be in each cell?
One.
All right. So look here now. So, right, so which means that these cells, they should be haploid, but it is actually diploid. So look at this now. Let us fuse it. So 3 chromosome is in this. Let's call it the sperm cell, right? So 1 in the egg as it should be. But instead of the male having 1, it actually have how much? Like, sorry, not 3. It has in 2. All right. So when this sperm with 2 fuses with the egg with 1, how much should be in here?
2.
The sperm of 2, the
3.
egg of
Oh,
1.
3. Sorry, 3.
Right. Now, how many should normally be in this?
2. Two.
Two.
Right. So 2 is the normal one, but with this junction, sometimes it might have less or it might have more. So in this case, how much it have?
More.
More.
3, right? We call it, if it have 3, it is trisomy. Trisomy. All right? 1 extra chromosome. So Down syndrome have extra chromosome. It's non, so the 2 you need to know about is Down syndrome and you have Turner syndrome. And it is as a result of this junction. Before we move, what is this junction? What is this junction? What is happening?
So this junction is when a cell can have less or more chromosomes when division occur.
All right. What is the main? That is partly correct. But what is the main feature of this junction? The chromosomes are not doing what? They are not dividing properly. All right. So remember, like with anaphase, they are supposed to separate, right? But if it does not happen, that is non-disjunction. They're supposed to separate, but they did not. So notice what they end up with is one cell containing the 2 chromosomes, right? And the other one, so this is in one instance where I'm showing you the one cell with both copies, which means that the next cell would be empty. And when it divide by a second meiosis, it would be empty. All right. So non-disjunction, it means that the chromosomes did not separate. All right. And because of that, it will lead to one cell having more chromosomes than it should, and the other cell have less chromosomes than it should. Now, to end this, meiosis of 2 stages, right? So sometimes meiosis 1, it can occur properly, all right? So non-disjunction, it can occur in meiosis 1 or meiosis 2. I know there are a lot of diagrams on the board. So what I'm going to do now, where this is non-disjunction in meiosis 1, all right? I'm going to show you that it can happen after meiosis 2. All right. Let me just erase some of this. Is anybody writing just before I erase? Just checking. I'm going straight up to 6, if anybody wonder, because I will have to leave right after. All right. So if you have to eat or something, you have to just wait or something. But yeah, I'm going straight to 6. All right, so meiosis 1, it will end with 2 cells, right? One set of chromosome up here and the next set down here. After meiosis 1, meiosis 2 will be just like mitosis, right? So I'm going to focus now. All right. Hope you can follow with this. All right. All right, so we finished meiosis 1. starting meiosis 2. This cell here. At the end of meiosis 2, what should happen? Basically, how should it end?
It must be haploid.
All right. We must get 2 cells. All right. One here and one there, right? Question. If this does not separate, split in, if this chromosome does not separate like this, how much would be in each cell? All right, don't answer that. This is what is supposed to happen. Good. This chromosome divides and end up with 2 haploid cells. The next option, so again, one cell has both chromosomes. This one have no chromosome. So what will happen after fertilization?
So which one is that?
Down here. So this cell, if it is fertilized, what will happen?
The zygote
No.
are gonna have one less chromosome.
Right. So again, this I'm just showing you that the trisomy situation, it can happen at meiosis 2 and meiosis 1. So this cell is diploid. So if you have a diploid cell fusing with a haploid cell, then you get a cell which is triploid, 3. Right? So this is supposed to give you 2 haploid cells. That is what is normal. But if it should happen that this disjunction occurred, meaning that these 2 chromatids did not separate, then it will lead to trisomy again, where this cell, if it fuses with a next cell, like a haploid cell, then it will have 3 chromosomes. All right? That is how you get trisomy, chromosome 21. That is why they call it trisomy 21. All right? So that's Down syndrome, Turner syndrome. Turner syndrome, it has less. So the Turner syndrome would be like if this one, Turner syndrome, this plus this. All right? I'm going to call it TS for Turner syndrome. This plus this Down syndrome, the extra chromosome. All right. So this is just to explain how you can get an extra chromosome inside of the cell. All right. So I hope that you understand it. For Down syndrome, it occurs on chromosome the 21st. So remember, we have 23 chromosomes. Trisomy 21. If you see the term, have you seen the term before? Trisomy. I just explained what trisomy means, right? You have a cell with 3 chromosomes inside of it. So if it says trisomy 21, it is referring to chromosome number 21. So it would not have separated.
Two less chromosome.
Please repeat.
So if it named trisomy 21, may ask if that means it have two less chromosome.
No, it have, no, remember trisomy, I want it to have 3 inside of it, right? So it would have an extra one. Monosomy, I want it, so this one up here is monosomy, it have 1 less. So 2 is normal. All right, so this is, right? This is trisomy. Okay. So when you hear trisomy 21, it is giving you, if the 21 is referring to the chromosome number. How many chromosomes, how many pairs of chromosome does human have?
How many pairs? Not
Yeah.
23.
Right. So with trisomy, right? How much would we have?
23 times 3.
No,
Oh, 23 plus 3, 26.
no, it's just, hold on, remember 2 is
24.
normal, right?
Oh,
Okay,
it would have been 47 in all.
Right.
Wait.
But in terms of pairs, it would be,
24.
right,
24, yeah.
yeah, 24 pairs. So it's just 1 extra. So the trisomy, a 1 extra chromosome you have. Yeah.
Wait, sir, repeat, please.
All right. So non-disjunction, it can lead to the cell having an extra chromosome or 1 less chromosome. So like with humans, right? We're supposed to have 23 pairs. So trisomy, it increases by 1. So it becomes 24. And monosomy, it will drop it by 1. Because you're losing out on a chromosome.
So monosome would have 22 chromosomes.
Right. Because if
Okay.
you notice, right, if you notice, after the second division, right, one of the cell is empty,
Yes, sir.
right? So when this empty cell fuse with a haploid cell, how much would be in that new cell? If an empty cell fuse with a cell with 1 chromosome, how much would be in the new cell? after the
One.
Right. And if a cell with 2 chromosomes fuse with a haploid cell, how many would be in that new chromosome?
3.
3.
Right. Yeah. So trisomy go by 1, monosome when it empty and fuse with a haploid one. So it decrease by 1. Yeah. So if you know, if them asking if you explain that, yeah, just it's never say simple, but just use all that you need to remember, right? Do not allow the cell, do not allow the chromosome to separate. So you're not doing the normal process of meiosis. So when it gets to anaphase, right? What is supposed to happen at anaphase?
They must be pulled to the opposite pole.
Right. So they must separate, right? And what does non-disjunction mean? What does
It
the term
means that
mean?
it
I
no,
don't divide in the correct way.
Right. So you're not going to separate them. So in meiosis, we are separating, in meiosis 1, you are separating the whole homologous pairs of chromosome. Disjunction would mean to separate them. So non-disjunction, we're not separating them. So you would put the 2 of these in 1 cell, and the next one would stay empty. That is if you are showing it at meiosis 1. If you're showing it at meiosis 2, this cell here would separate to give you 2 diploids, right? And then now for meiosis II, this one of them, it will not separate. So one cell will be diploid and the next one will be empty. All right, that is non-disjunction. It can happen either at meiosis I or meiosis II. Anaphase is the problem, all right? So with anaphase, it should not occur. So if you're showing it in meiosis I, anaphase should not take place, as anaphase is where they separate. Is that clear? So just remember that if you want to show non-disjunction, do not carry out anaphase. Well, yes, don't carry out anaphase, but that don't mean this. When I say don't carry out anaphase, that don't mean you should not divide the cells. The cell will divide, it's just that the chromosomes will not go to opposite parts of the cell. All
So
right?
just show metaphase alone
No,
and
man.
prophase.
I will show the prophase, the metaphase. With anaphase, it comes out to one side. Do not pull them apart.
Wait till you just freeze up.
All right, so I'm gonna say, when we say don't show anaphase, well, yeah, so I guess skip from metaphase, or if you want, even if you're trying to show anaphase, put the two chromosomes one end of the cell to show that it is not separating. All right?
Sure, because in an anaphase, it basically I pull them apart evenly.
Right.
And that's
On a non-disjunction, it's not supposed to pull apart. So apart from that, the final thing, remember meiosis has 2 stages. So if it doesn't, if you do anaphase in stage 1, do not do it in stage 2, right? So because meiosis have 2 stages, you have 2 sets of anaphase. So you can show it at stage 2 or stage 1. So if you show it at stage 1, do not show it at stage 2, right? So yeah. Let's look at some variation. We started well on variation. What I want to get to, where was it? Disruptive, stabilizing.
Sir, they always tend to ask about the